ta'ala wa barakatuh. Today I want us to look at the topic Chapter 3 Biology for what that is the cell. Now let's look what the cell means. We divide the cell. We're going to see which instrument we use to view the cell. Now the cell basically means it is the basic structural and functional unit of an organism. Having said that the cell is the basic structural and functional unit of an organism, now I want us to look how is the cell a basic structure, as well how is the cell a functional structure. Let's start with the basic structure. For example, when we have a living organism, that living organism is made up of organ systems. From those organ systems, they are being made up by organs. From those organs, they are made up by tissues. From those tissues, they are made up by cells. Now we have the cell. We have tissue. We have organ. We have organ system. And finally, we have the organism. Now, you can see that there will be no tissue without the cell, and there will be no organ without the tissue. An organ system wouldn't be existing without the organs, and the organism will not be there without the organ system. And therefore, in essence, this can be linked up and say that the cell is the basic structure from which all these other, the tissue, the organ and the organ system arise from. This is now basically a structural basic, such that the tissue will not be there if there is no cell. Now, the next thing I want us to look at is how is the cell as functional structure. For instance, let's look at tissue like the blood tissue. The blood tissue is made up of the fluid part of the blood, that is the plasma, and it's also made up of the cellular component, that is the white blood cells, the red blood cells, and the platelets. Each and every cellular component, or rather the fluid in the blood tissue, is functional at its independent state, and therefore the blood tissue cannot work as a whole at once, but is only functional at the cellular level. Good. Now, another very important thing is that the cell is a microscopic structure. Point to note. The cell is a microscopic structure. The cell is a microscopic structure. If you say that the cell is a microscopic structure, simply means that the cell is very minute structure that cannot be viewed, or rather cannot be seen with the naked eye. And therefore, a powerful magnifying instrument should be put in place so that the cell organized, or rather the structures found within the cell can be seen. And those structures and those microscopic lenses are of two. We have two types of microscope that you're going to be using. We have the light microscope, and we are going to have the electron microscope. The light microscope. Those are the electron microscope. What's the difference between the light microscope and the electron microscope? The light microscope is entirely dependent on the light from the sun to illuminate the specimen, whereas the electron microscope uses beam of electrons to illuminate the specimen. And therefore, if you require finer details or finer structures of the cell, the best instrument or magnifying instrument to be used is the electron microscope. Now, similarly, these are just microscope, but what differs is the way in which they magnify the object. This one has higher magnification than this. And again, the electron microscope has higher resolution than the light microscope. Now, the light microscope and the electron microscope, they work on two principles. Principle number one they work on is that they do something called magnification. Magnification is the enlargement, or rather increasing the size of the object by multiplying by a certain number. That is, for example, you can magnify an object times 10 times or times 20 times. We also have another magnification. Basically means the ratio of an image size to the object. And what you know is that the object is bigger, the image is bigger than the object, and therefore the magnification will be having for example, let's say the image was having 10 and the object was 2. Now the magnification will be times 5. The magnification is the ratio of the image size to the object size. Now let's have another term called the resolution. Resolution, on the other hand, basically means ability of the microscope to distinguish two close structures as distinct entities. At some point, if you keenly look at these structures, they appear as one on their close proximity. And therefore, with the use of a powerful magnifying lens like the electron microscope, these two structures can be viewed as distinct or rather separate entities. Now, from there now let's look at the parts of the microscope, and their function, parts of the microscope. Part of the microscope, we have part one, we have the arm or limb. The arm or the limb holds body tube and the stage, and the stage holds body tube and the stage. We also have the body tube itself again, body tube itself. The body tube holds the revolving nostrils, and it also contains the eyepiece lens as well. Now let's look again. What is the revolving nostrils? Now, the revolving nostrils basically it holds the objective lenses. By the revolving nose holding the objective lenses, it thus enables them to change from one objective lens to the other. Now, next, let's look at the objective lenses. The objective lenses contributes to the magnification, We also have the eyepiece lens. The eyepiece lens also contains a lens that still contributes to the magnification. Of the object. Now again, we have the diaphragm. The diaphragm, it regulates the amount of light. Basically, it regulates. The diaphragm regulates the amount of light passing through the condenser. We also have the condenser. It concentrates light onto the specimen on the stage. Concentrates light onto the specimen on the stage. Again, we have the mirror. The mirror reflects light through the condenser to illuminate the specimen on the stage. The mirror reflects light through the condenser to illuminate the specimen on the stage. We have the cause adjustment mode. Cause adjustment mode. This one raises or lowers the body tube for a longer distance to bring the image into rough focus. We also have the fine adjustment knob. Fine adjustment knob. The fine adjustment knob class, it raises or lowers the body tube for a shorter distance to bring the image into sharp focus. We also have the stage. The stage is the platform where the specimen, it is the platform where the specimen on the slide is placed. Platform. Where the specimen on the slide is placed. We have the clip. The clip holds the slide with the specimen in position. Holds the slide with the specimen in position. It holds the slide with the specimen in position. And I think these are some of the parts of a microscope. Now, from there now, the microscope has some handling and care. What are some of the handling and care of the microscope? How are you supposed to be handling the microscope? The microscope is a very delicate and expensive instrument, and therefore it should be handled with utmost care, because once it is handled, for it to be restored back, it is very expensive. Again, it's a very delicate structure. Therefore, what are some of the things? When carrying the microscope, you use both hands. One hand should hold the arm, while the other one should be holding the base. Another important thing about the microscope is, in a working bench, never place the microscope too close to the working edge, so that it may fall off and break. Number 3, do not touch the mirror and the lenses with your fingers unless you identify them. When cleaning the microscope, use a soft tissue to clean it. And that is some of the care and handling of microscope. There are very many, but just those are just few. Now, how do you use the microscope? The microscope has its own uses. For example, the mirror should be in position such that it should be able to receive the light, so that it can illuminate the specimen on the stage. The objective lenses should be clicking into position. Very good. Those are some of the uses. Now let's look at the cells seen in analyte microscope. Structure of animal cell. Structure of an animal cell under light microscope. Now, the structure of an animal cell under light microscope goes like this. Good. Structure of a plant cell. Structure of a plant cell under light microscope. Structure of a plant cell under light microscope. Now we have this structure like this. Very good. Now let's start labeling the parts. This is the structure of an animal cell under light microscope, the structure of a plant cell under light microscope as well. Now this one is called the cell membrane. This one is the cytoplasm. This one is the nucleus. Let's look at the plant cell. They have cell wall. You have the cell membrane. They have the nucleus. You also have the cytoplasm here. You have the tonoplast. You have the subvacuole. You have the chloroplast as well. Now, this is the structure of an animal cell under light microscope, and this is the structure of a plant cell under light microscope. The structure seen are the cell wall, the cell membrane, the nucleus, cytoplasm, tonoplast, subvacuole, and chloroplast. Now let's look quickly the function of each and every structure. Let's start with the cell wall. The cell wall. What is the composition of the cell wall and where it is located? The cell wall is the rigid outer covering in plant cell. Rigid outer covering. This is a rigid outer covering. In plant cells. The cell wall is the rigid outer covering in plant cells. Again, it is composed of cellulose. Having said that it is the rigid outer covering in the plant cells, the cell wall is also composed of cellulose. Again, functions of the cell wall. Functions. Function number one, it gives the cell its different shape. Gives the cell its definite shape. Number two, provides the cell with mechanical support. Number three, it prevents mechanical injury of the cell. Prevents mechanical injury of the cell. Now we are done with the cell. The next cell organelle is the cell membrane. Cell membrane. Sometimes called plasma membrane. At times called the plasma lemma. All these they just refer to the cell membrane. Now, cell membrane, it is found in both plants and animal cells. It is composed of two protein layers. The cell membrane is composed of two protein layers and a phospholipid layer. The phospholipid layer in the structure of cell membrane is sandwiched between the two protein layers. Now, what are the functions of the cell membrane? But before function of the cell membrane is function, it encloses cell content, The cell membrane, first one is enclosed cell content. Number two, it is selectively permeable. That is semi-permeability. It's selectively permeable. Whereas the cell wall, it is entirely permeable. The cell wall is entirely permeable, whereas the cell membrane is selectively permeable, rather semi-permeable. Now let's go to the next one. That is the nucleus. Next structure is the nucleus. Nucleus, it is the largest cell organelle within the cell. Having said that it is the largest cell organelle within the cell again, the nucleus contains various structures. For instance, the nucleus contains structure like the chromatin. The chromatin. Chromatin contains the hereditary structures. Again, the nucleus has the nucleolus. The nucleolus manufactures ribosomes. Again, the nucleus has pores called the nuclear pore. That's the nuclear pore. The nuclear pore. The nuclear pore is the where the materials move in and out of the nucleus. The nuclear pore. From there, what's the general function of the nucleus? The nucleus controls activities of the cell, controls the activities. The nucleus controls the nucleus. The general function of the nucleus is that the nucleus controls the activities of the cell. What have we said the nucleus? It contains hereditary structures, that's the chromatin. It also contains the nucleolus that manufactures ribosomes. Now, both the chromatin and the nucleolus are suspended in the fluid of the nucleus, that is the nucleoplasm. The nucleoplasm. Both the chromatin and the nucleolus are suspended in the fluid of the nucleus, that is the nucleoplasm. And the nucleus has a membrane, that is the nuclear membrane. The nuclear membrane encloses the nucleus, and therefore it has pores and openings through which materials move in and out of the cell. Now, what is the general function of the nucleus? It controls the activities of the cell. Generally, there is a brief information about the nucleus. Let's go quickly whatever remaining. We have the cytoplasm. The cytoplasm is a viscous liquid. The cytoplasm is a viscous liquid in which other cell organelles are suspended. It is a viscous fluid in which other cell organelles are suspended. If you look at this structure, this is now the cytoplasm. The cytoplasm contains a fluid in which other cell organelles, the chloroplast, the nucleus, the subvacuole, the tonoplast are suspended in it. Now, what's a viscous fluid? A viscous fluid is a fluid that is not easy to pour out. For instance, okay, maybe I can say a honey is a viscous liquid, is more viscous than normal water, than distilled water. The viscous is that how the molecules of that particular fluid are close to one another and are sticking to one another. Now, what is the general function of the cytoplasm? Cytoplasm, it is the site function, site for cellular chemical reaction. Site for cellular chemical reaction. Now, let's go quickly. From there, we go to the chloroplast. The chloroplast is an oval-shaped structure found in plant cells. It is the site for photosynthesis. And lastly, the subvacuole, it contains a fluid that is the cell sap. The cell sap contains sugars and salt. The sugars and salt, they regulate the osmotic properties of the cell, and therefore the subvacuole is being enclosed by the chloroplast. Thank you very much, and in our next lesson we will be looking at the cell organelles seen under electron microscope, where finer details of the cell structures will be displayed. Thank you very much.